day 2 of our intro course. And today we're going to be talking about cell lines and start introducing you to the final project. So in case you're lost, hopefully you're not, but let's go over what this course is about. Um, so basically we're going to go in depth from what was seen in the crash course, breaking down each step that we introduced. And there's also going to be a final project that will have a little fun competition for the last day. So I'm Emily again, if you didn't make it to the crash course. Sangeeta's here as well, but the other instructors you'll see along the way are Adil, which you might have met in the crash course, Jasmine, and Megan. So to go over our syllabus, we've had this week, which is the overview on Monday. This week, this lesson, we're going over cell lines. Next week, we're going over media and scaffolds, then bioreactors, downstream processing regulation. We're also adding to that marketing, because we notice we have a lot of Sloanies in the audience.
Um,
And food science, and then we're going to have our final project presentations taking up all of our last session. So let's introduce the final project. We have a couple of different options depending on your circumstance. So option A would be to show off your homemade recipe or fermentation that relates loosely to food science or cell ag. It's kind of hard at this point to make your own cultured meat, though you could find there are some recipes on Shojin Meats DIY that you could try those out. But what could be a lot easier is making your own sourdough, cheese, kombucha, yogurt. So that could be fun. There's also things like alginate caviar and biomaterial bacon, where you can sort of get the feeling of what would alternative protein look like. Option B would be to make a variation of an existing paper. So we talked last session about how you can decellularize leaves to create a scaffold. So you can make a decellularized salad. You can do a consumer survey, or even a survey of topics like consumer acceptance or research acceptance at MIT, or in your relevant college or university, or another social topic, or you can make a review paper on any topic of your choosing. But we really hope that MIT students, undergrads, can make a project proposal for a year off. So this is an option that we're especially excited about, because we are working with the BE department, the Course 20 department, to start getting UROPs into labs to work on cellular agriculture topics. So this project would include, this project would be a proposal. So we'd like you to find a potential principal investigators, and we'll help you email them once you're done with the proposal. Give background, materials, methods, timeline, and some relevant papers. So our timeline for the project is you're going to start working on the background and research today. On Monday, you're going to make a working abstract and start planning and ordering your materials if that's what you need to do for option A or B. Wednesday, you can start researching possible professors, keep planning and ordering your materials. Hopefully they'll arrive by Monday of week 3, and you can start your hands-on portion of your project, or start writing your final draft of the proposal, then wrap up your project in week 4 and present on Wednesday. And I said that there was going to be a little competition for the presentation. So the winner will be decided by popular vote, and the winner will be getting the Perfect Day ice cream of their choice, made by Brave Robot. I've heard that some people have already had this treat. I have not. I'm going to be so jealous when you get it. So are there any questions before we begin? We'll be talking about the final project later, but yeah.
Yeah, I have a quick question because you said that it sounds like the UROP proposals, the third one is geared mainly towards MIT undergrads. Does that mean that if you wanted to do that one, you should probably pair up with other MIT undergrads, or how would that look? Or is this also completely individual? I don't actually know how that.
I would say actually for if you are looking to propose a UROP, I think it would be better if you were working by yourself because UROPs are
Okay.
typically by yourself.
Yeah.
But yeah, if you had a team member, perhaps you could do somewhat like related projects and help each other. Option A and B, where you're making more hands-on experiments, maybe if you have a roommate or people that can help you, you can transfer things over. That might be helpful, but we've designed this so that it can be individual.
Okay, thanks.
Cool. Anyone else? We have some people joining us. Hello, welcome. This is welcome to day 2 of the intro course. So we just went over a review of the course. So if there's no further questions, we can go into today's content, where we're talking about how to find yummy cell lines. So as we saw in the crash course, cellular agriculture can be split into precision fermentation and culture and meat. I know that I went much more in depth probably into culture and meat than in precision than we went into precision fermentation last time. So here I want to address precision fermentation first. So precision fermentation uses microbes as factories to produce functional ingredients that they may not otherwise produce. So this can be made by like inserting basically a piece of DNA into a microbe's genome, and that makes it so that they produce the protein product, or they produce enzymes that allow them to go through the biosynthesis pathway to make fat or some other molecule that isn't a protein but is produced by proteins. So the cell lines that we have for that, what are microbes? Microbes are yeast, it can be other fungi and bacteria. So there's not a lot of research into precision fermentation yet, but what's a really interesting read is Perfect Day's GRAS application, where they talked about how they're using Trichoderma ricei. I don't know if I'm pronouncing that correctly. But this is a fungi that has been proven safe already in making several food grade enzymes. So it made it quite easy for Perfect Day to get approval that their whey protein was also safe. So attributes you want to select for when you're looking at precision fermentation is how much protein or any of the desired product that you want, how much can you extract from that cell? How easy is it to extract the protein from that cell? Because in some cases, the cells might be secreting it already, or you might have to go in and sort of burst the cells open. So how easy is that process? What is required? You want to make sure, as Sangeeta said on Monday, that there's generational stability. There's not a lot of mutations going on. You want to make sure that your microbe is safe, so it doesn't have a lot of secondary metabolites or toxins. And especially you want to make sure that your end product doesn't have anything that you don't want. It can just be 100% pure. You also want to have minimum consumables. So looking at cells that don't have a lot of specialized media requirements would be great. And can they grow well at scale? And we're talking like 20,000 liters when you think about at scale. And a couple of ways that we can do this is we can look at genetic engineering, as Sangeeta went over last time, or adaptation or breeding. So I thought what Sangeeta said about the E. coli that is typically heterotrophic, but through directed evolution is made autotrophic, so it can make its own food just with CO2, was super cool. And this shows that you don't need a lot of genetic engineering for this sort of thing. Microbes are quite resilient, and a lot can be done just with breeding and evolution. So to make this a little bit interactive, can anyone give me some guesses for what DNA we should insert to produce our desired product in precision fermentation? Like what elements would you need?
Hi, I can try it. So you need the gene that, sorry, I don't speak English very well, so I'll try the gene that codes for the protein, so the open reading frame of the protein. And then you need the promoter, which is needed for the expression of the gene. And then you need to terminate the sequence of the gene. And yeah, I don't know if it counts because I study biotechnology, but that's what I put.
Yeah, so you covered just about everything I would. The one thing that I would add would be to have some sort of selection marker.
Yeah, sure, you're
so
right.
that you're proving that you have mutated your cells correctly. Yeah, and so that was a great answer. One thing that you might think about is it might not be the exact same sequence as you would see in a cow, because depending on the microbe, that microbe is going to have different ways that it processes from gene to protein. So Perfect Day doesn't actually use the exact same sequence as seen in a cow, that they used a codon optimized sequence for their fungi. And though it's not the same genetic sequence, it still produces the same protein, which is what matters at the end. So then a final question as we go into precision fermentation, do you all think that this is genetically modified food if we're talking about products, proteins, other molecules that we're getting from these microbes? You don't think so? I see some shaking heads from Fernando.
I think it kind of depends. Like if you're eating, if like the microorganism is part of the final product, like you're eating, like, you know, you're growing an entire colony of fungus or whatever, and like that mushroom or you know, that fungus is the final product, I feel like that's kind of genetically modified. But if it's more of like the catalyst, like a sourdough bread or whatever, to creating flavor and it's more for the byproduct, then I think you could make the argument that it's not.
Yeah, I think that if what's genetically modified is the organism. So if I'm not eating the organism, I don't think that the food is genetically modified. I don't think that saying that food is genetically modified doesn't make any sense to me. I mean, maybe with vegetables, but with precision fermentation, that doesn't make sense.
Mm-hmm.
Yeah, I think it just really depends on how specific you want to be with your criteria for what's genetically modified. And if, I don't know, I could definitely see an argument though, just to, I guess, play devil's advocate for the other side, that if, one general, very, very general definition would be if genetic modification is involved at any step of the process, then as genetic modification being like, going in, like inserting sequences and stuff, then maybe that's genetically modified, even if the final product, there's nothing that in itself had like, there's no living, you're not eating a living or previously living organism directly that was genetically modified.
Rachel, raise your hand.
Yeah, I think there's no doubt in my mind that this is a genetically modified food. but I think that there's a negative connotation with genetically modified, and I don't think it's problematic at all. I think when you see base substitutions, that's the same thing that all the RNA vaccines for COVID are using. It's just, this is how we do it. There might be some slight differences in post-translational modification, but you have to be okay with that. That's how science works. That's how medicine works. People are getting this all the time, but for some reason, when it comes to food, it's problematic. And typically you see people have issues when like certain genes are inserted in food that help with like bacterial resistance or things like that. And all of a sudden a soy protein gene is inserted in a vegetable and someone has a soy allergy and doesn't know. So that's like when it's problematic. But I think there should be some sort of distinction, and to the general population, it's confusing. But in my mind, back to my original point, this is absolutely genetically modified and is not a problem.
I mean, so is it genetically modified if the cell is not part of the product that people are eating as food, or is it is there a difference if it's just the protein or just a molecule, so it doesn't have any genetic elements in it?
Yeah, I still think it is, because I think that it's coming from a different organism and there was some sort of like differences in sequencing. Now, while that might not fully change the protein, I still think, again, those like post-translational modifications might be slightly different. And then with regard to like, yes, it's still all the same kind of codon, or it's going to code for the same amino acid, so it might actually be the same. But we've even seen some cases that, like, I guess whether or not it sticks around longer really doesn't affect the actual protein that's produced. But I think there's some, maybe it's not genetic, but I still think it's some sort of modification that should be noted.
Well,
It's not
I'm
natural.
actually, I don't know, maybe you know this, Emily or Sangeeta or someone else, definitely chime in. Is it the case that I guess whoever officially, whether it's the FDA or somebody else, the ones that label things as genetically modified or not, if it's say it's a glass of milk that came from a genetically modified cow, would that be a genetically modified food? Because I see that as being functionally equivalent to like the constituent and constituent proteins and enzymes coming from a genetically modified, essentially bacterial or fungal factory, and then you just kind of like engineer a glass of milk that way. It seems like it's the same thing. So would a cow that's genetically modified produce genetically modified milk?
Yeah, I don't know. I'm not. I don't think that Perfect Day has gotten the certification yet to show that whether they're genetically modified or not. But this is I've asked this question because it seems to actually be a debate between companies. So Perfect Day and Geltor both call their products GMO free. But I think that's a bit of a misnomer, because GMO means genetically modified organisms, and if there's no organism in the final product, then of course it's going to be GMO free. But it might be slightly different to say, is it genetically engineered? But Impossible actually
Exactly, yeah, because a glass of milk that comes from a genetically modified cow, like it's not what is it exact? It's hard to say what you would even genetically modify in that final product glass of milk, but yeah, exactly. But there was involved in the process. There was something that was genetically modified.
Yeah, but on the other hand, Impossible has come out and just even says on their website that their products are genetically engineered, and they argue more that like we need to stop making GMOs such a horrible thing. There's ways to do it responsibly. For instance, my personal opinion is I think that the line gets a bit more fuzzy. I think that if the cell isn't involved, then I don't think that is a GMO. If it's the exact same protein with the exact same characteristics, post-translational modifications, I feel like that's not genetically engineered for me. But I think that there's a different line, actually, when you talk about Geltor's products, where they are not talking about necessarily collagen that's found natively in cows or in any existing animal, but they're actually designing collagen that has the specific properties that they want for a particular application. Yeah, we have more answers in the chat.
I had just shared a photo, and I think Rachel had said, I like genetically engineered, but
yeah,
I was making pasta the other day and I noticed this in the jar, and it's very normal ingredients, but it says, yeah, if you want to pull up the nutrition label, like on the back of the jar kind of thing.
Yeah. Is this it or
I think it's like
this one?
Yeah, that
second.
one.
Yeah. So right there underneath the ingredients, it says the ingredients from corn, soy, and sugar in this product come from genetically modified crops. So I thought that was an interesting way of, I hadn't seen that before in a food label, and let alone a pesto bottle, but
Yeah.
yeah. Just something to know. But yeah, the sugar and probably the corn products they're talking about are not different from those that we had used before, just kind of similar to fermentation.
Mhm.
But probably the gene edits that have been done in this case are just to make the plant grow better.
Yeah.
There you go. So it looks like they have some kind of system in place, at least to address this interesting middle ground. Maybe. I don't know how standard, yeah, because I've never personally seen that before. That's very good. That
Yeah.
works.
And I think it's going to be different depending on what country you are thinking of how much this is going to be a problem. I think in the US, we're pretty lenient, but the EU has much stricter standards on what's genetically modified, and if it's genetically modified, can you make it into a food? Yeah. Okay, that's cool. So now that we talked a little bit about precision fermentation, we can go into cultured meat. So cultured meat, as we've learned, is meat that's produced outside of an animal through cell culture. And so you're taking a sample of cells, growing it up into a bioreactor in a scaffold, making a meat product. And we went over last time this diagram of like every step of the way. So today we're going to really be talking about muscle sampling and these isolation or preservation of these cells. So to give an overview of last time we talked about how when you're thinking about where you're getting your sample of cells, there's a few characteristics to consider. You have to consider where you're taking the cells from. Is it the front shoulder, the back shoulder, where have you, depending on the animal, there's going to be a different concentration of each cell type. You're going to have to think about the age. So the older the animal is, typically, the fewer muscle stem cells they have, for instance. As you can see here, for humans, your concentration decreases dramatically just from when you're born to when you're 18. And then also it might be relevant, like the breed of cow, the sex, and the conditions that the animal is in. Is it under a lot of stress? What is it eating? Is it going to be outside, getting lots of exercise? So this is all important. Then next, you're going to have to think about your methodology for biopsy. There are different ways that you do it depending on the cell types that you're looking at. But for muscle, you're going to take a needle or a scalpel to go into that muscle tissue. But before you do that, you're going to have to first fix the animal in place and give them anesthesia. So when you think about animal welfare in this process, the most stressful part of the experience is probably going to be before they get anesthesia. So when you're fixing them into the cage and giving them the shot of anesthesia, that's going to be when they're under most stress. So to minimize the amount of stress that the animal goes under in their lifetime, it would be wise to take several biopsy samples per session, and also have at least 1 month of recovery between sessions, and have a slight variation in location from session to session, both to allow the animal to heal so they feel better, but also so there's not a lot of scar tissue building up and you're not getting the cell types you want. So then we talk about what cells do we want to isolate. In the crash course, I'm focused mostly on muscle cells, but there are actually a few more types that you might think of. Anyone like shout out some other types of cells you might want for your cultured meat?
Fat cells.
Yeah. There we go. And there's one more, but you might not think about it.
Bone?
Bone might be, yeah. If you wanted like cartilage specifically,
Yeah.
yeah, you
Right.
might use for a sort of scaffolding. Yeah, I'm actually not going to talk about that, but there is one other type that when Adil was talking about media formulations.
Fibroblast.
Blood cells.
Okay, blood cells also. Yeah, you can have cardiac cells that help with oxygen flow. But what I'm actually thinking about is feeder cells. So those are liver, placenta, or what is the third one? Pancreatic cells that can produce some growth factors to replace expensive media. So to go over from last time, we talked about muscle cells. So you can get muscle cells from a few different places, like embryonic or induced pluripotent stem cells or mature myocytes. But the way that's been used, I think the most in this industry is muscle stem cells, which are adult stem cells present in skeletal tissue, which can self-renew and are used for repair of mature myocytes or muscle cells. But then when you talk about adipocytes, it's a much newer field, even within cultured meat, which is a new field. We've only thought about in the past couple of years that we actually need to have some juicy fat cells in there. So there are a couple of different options that we could have. You could take mesenchymal stem cells, which can actually produce muscle cells as well, or myoblasts, but the process to make sure that they differentiate into adipocytes is pretty well defined. Adipocytes are basically fat cells. You can use them interchangeably, sort of, but they don't mean the same thing. Like fat cells or adipocytes are not your only source of fat, but they're sort of the main component. So mesenchymal stem cells are pretty cool. We know how to get them, but unfortunately, they don't have unlimited proliferation capacity, even as stem cells. And it especially unfortunate is that their capacity to become adipocytes actually decreases as the cells reach their proliferation limit. So you're going to run into this sort of balance of how much can you produce at scale and how much of that is efficiently going to become adipocytes. So then you have DFAT cells, which are basically made by de-differentiating mature adipocytes in culture. It doesn't require any genetic modification. It's just done by culturing the cells, and they revert back to a more stem cell-like fate. And this can also be differentiated back into adipocytes in a fairly well-defined protocol, but it has a much longer-term proliferative capacity. In addition, you have the committed pre-adipocytes. So these are cells that are already committed to adipogenesis. They're not going to become muscle cells or bone, cartilage, any of that. And then we're going to talk about those feeder cells. So these feeder cells can be liver, pancreas, placenta, but they secrete growth factors that can be circulated to the primary cell tanks in order to be basically a replacement for the media. And this is kind of a really cool process that a couple of companies have gone in on. So IntegraCulture's CoolNet and Fork and Goods bioreactor systems both use this process that basically is supposed to mimic the body, where you have different organs mimicked by different tanks. And you can have a bunch of different configurations depending on what cells need each other to survive. But basically, it allows you to think about the media formulation as sort of a black box, because you know that these feeder cells produce the growth factors that muscle and fat need to survive in the body. So you don't have to think about any complex media formulation, as well as you don't need to then buy those ingredients to make those growth factors. And then we talked last time about a protocol for cell isolation, so I won't really go into this, but basically you cut up the tissue, you do some sort of enzyme dissociation, filter and centrifuge to remove debris and isolate just the cells in your biopsy sample. And then you can isolate the cell types you want based on different cell sorting methods like FACS or MAX.
Emily, I think we have a question from
Yep.
Elizabeth.
Are
Oh, it
feeder
was
cells
just-
a replacement for media, or would feeder cells just supplement media? Yeah, so they wouldn't be, I guess I didn't misspeak there. They're not going to be a replacement for media, in that you're still going to need like your simple sugars. So you're still going to need those nutrients, but it could be a replacement more for those really expensive growth factors
Okay,
that
so like
make up the majority of cost in media.
something like serum. It's like a replacement for the fetal
Yeah.
bovine serum. Okay,
Yeah.
cool.
All right, so we talked about those 3 cell types, but we mentioned over and over again that it's going to be, you have to really think about their proliferative capacity. So one way to get over proliferative capacity is to just not deal with primary cell cultures. So any of these native cells, they're going to require you to come back to an animal over and over again. So there's going to be batch to batch variation, and you're still going to have some amount of animal farming, which you may or may not have a disagreement with. So what you can have instead is cell lines. So cell lines are cells that for one reason or another have infinite proliferative capacity. But there are several ways that our bodies have designed ourselves to not be immortal. So we have several cell cycle checkpoints that we go through each time we have a cell division. Each time you have a cell division, your telomeres are going to be shortening a little bit, and eventually the telomeres are the ends of your DNA chromosomes that protect your DNA from damage. So once they're shortened enough, your actual vital DNA is starting to get damaged, and that will activate the senescence pathway, where your cells will just stop going through cell division and enter the senescence state. And without these cell cycle checkpoints working correctly, that often is what leads to cancer, because cancer is basically infinite proliferative capacity. So there's a couple of ways that we can make immortalization of our cells occur. One, we can just not do it ourselves. We can look for it in nature. So the HeLa cells from Henrietta Lacks were just found from her cervical cancer. We can also make it ourselves by expressing telomerase, which repairs the telomeres, but is typically only found in the embryo. Or we can inactivate these cell cycle checkpoints. So we can repair our telomeres by expressing telomerase or having telomerase in the cell culture mixture, or having some plasmid where telomerase is expressed. Or we can infect our cells with different viruses, such as SV40s or HBV, that have actually been evolved to make their host cells immortal. So SV40 is believed to, in nature, infect muscle cells that are senescent. So they've found a way through their large T antigen to turn off their host cells checkpoints so that their host cell can become immortal, go through these infinite cell divisions. And each time the host cell goes through a cell division, SV40 virus also goes through a cell division. So we can infect our host cells with these viruses, or we can just insert the genes that are important for this immortalization process. So for SV40, that's the large T antigen. Oh, I didn't go over here, but what you can also do is just knock out the checkpoints themselves or put them under some new regulation. So that would be really cool, but that is quite a obvious genetic modification. This is definitely a genetically modified organism, but there are ways that you can actually kind of reverse your modification. It's called a footprint free method. So these are systems of genetic modifications where you can actually add an enzyme later in the process, and that removes the genetic modification itself. So the final cell product does not have any sort of modification at all. There's no foreign DNA. So Wild Type, which is a cultured meat company, has actually patented one example of a footprint-free method where they've introduced modifications to prevent differentiation and promote proliferation. But these can be removed later by enzymes called recombinases that identify the specific sequence of these modifications, cut it, take it out, and stitch it back so that there's no modification left in the final product. So that might be really cool, but cell lines do have their own limitations. Number one being that there aren't a lot of them available right now, especially for agriculturally relevant cell lines. We don't have really any so far that for muscle cells, because there has been just historically limited funding for cell line repositories. So researchers often have to first make their own cell lines before they can start doing any cultured meat research. And GFI, the Good Food Institute, as well as New Harvest, are funding several projects to start making these repositories for cultured meat to lower the barrier for cultured meat research. It's also quite difficult to establish a cell line. There are kits that you can buy, but it's not going to work every time. There can be crises that the cell goes through during the immortality process that cause the process to fail, especially cell lines that we don't, cells that we don't know a lot about, like shrimp, for instance, have been very hard to immortalize, and we actually don't have any immortal cell lines at all for most seafood for most animals that we use for seafood, because the biology is quite different from the mammals that these kits were optimized for, and we just don't know enough about their physiology. So then once you have that cell line, there is going to be some genetic drift over time just by like random mutations that happen. And there's also going to be cross-contamination. So talking about those HeLa cells again, a few years ago, there was sort of this shock throughout a lot of biology industries when they found that almost every cell line that they had been infected with HeLa cells along some point. So you need to find some way to confirm your cell's identity before you put it into culture. And the best way to do this is to have repositories where you have frozen stocks of that specific cell line in place, and you can compare, you can confirm their identity then before you put it into a larger culture. So I talked about that the capacity of cell lines that we have so far is limited. Actually, the only muscle cell line that we have right now for mammals is mouse. or the C2, C12 cell line. And this is the cell line that most cultured meat researchers are using right now, such as Stephanie Koecki has that little muscle meat, mouse meat in her Petri dish. So this isn't a great solution. You can consider whether or not consumers would want to eat cultured mouse meat. So we really do need these agriculturally relevant cell lines. I'm gonna look at some questions. Okay. I want to try mouse meat. Yeah, robust cell lines. So I would say when people are talking about robust cell lines, they're talking about is it stable? Is there going to be, or is there going to be a lot of mutations at a very high rate? Also, is it going to be, like, does it require a lot of babysitting to culture, or can it, is it quite resilient, like at different temperatures, at different culturing conditions? Does it need a lot of crazy media requirements? Is this session also being recorded and uploaded? Yes, I'm sorry, I didn't say that in the beginning. All of these sessions are going to be recorded because I do know that we have a lot of busy people who are doing part-time jobs or research, and people from throughout the world, different time zones. I have one small question about key functions to decide cell line quality, good or bad. Immortality is one of the key functions, but anything else for culturing meat applications? I'm not sure. Okay, so when we think about cell line quality, I think this question is asking for what attributes you would like. So immortality is a good one, or otherwise high proliferative capacity. But especially when you're talking about cultured meat, you want to make sure that the cells are also safe to eat and that they're yummy, they're tasty, because there's a sort of basal flavor that comes from the cells themselves. And Megan will talk about that some more in her food science talk later in this course, I think on week 4. Yeah, but we want to make sure that it's safe, that it grows well, and that it's tasty, that people want to eat it. All right, so
I think there's one more question actually at the very top from Tim. It's, do the current criteria for genetically modified only focus on the end product then? Or as long as it's the same at the end, is it generally agreed that it is not a GMO? I think this refers to the footprint-free.
Yeah. I think it'll depend again on what country you're looking at. I am familiar with the US system, and I think wild type was built for the US, and I don't think it would count as a GMO for the US at least, but in Europe it might be different, or China or elsewhere. Yeah. Okay. There's no more further questions. I'm going to talk a little bit about what we could look for in cell lines in addition to just being agriculturally relevant. So for the first off, we could have alternative exotic cell lines. So we could think about animals that we can't typically farm and just do some hunting for what meat would actually taste the best, rather than think about can we domesticate and farm this animal in large quantities. So Vow Foods is sort of the spearhead company for this. This is a kangaroo dumpling that they've had, but they're also working on cell lines for lion and tortoise and yak. You can also think about insect cells, which might not be so yummy for Western consumers, but there are many cultures that do eat insects. So I'm from Texas, and in Mexico, there are several dishes that use crickets. So they're somewhat agriculturally relevant depending on where you are, but they're also useful from a bioengineering standpoint because insect cells don't require the amount of heating as mammals do because they are cold-blooded. They also like they also like don't need as many growth factors as mammals. So they could potentially be much cheaper to cultivate, and both from a cost and an environmental standpoint. So then we could also think about enhanced cells. So last time I talked about vitamin A enhanced meat from Andrew Stout, but he's also thinking about omega-3s, therapeutics. There's also like color and flavor compounds that you can add, as well as modifications that you can do to the cells to just make them grow better in culture. So for instance, there's one company working on that has a patent to modify their cells so that they can turn ammonia into an amino acid, so that ammonia is a very common metabolite for cells and it's toxic to cells. So it can be quite problematic if you're thinking about a large culture. And in that modification, you can turn this toxic byproduct into a nutrient for cells. And then, of course, as we mentioned earlier, you really have to think about taste when you're selecting for these cell lines. Cool. So that's our content for the day. If there's further questions, I can take them now, or we can talk about our homework and our final project. All right. If there's not questions, then I
actually do have,
yeah.
sorry, I have a real quick question.
Yeah, go ahead.
In general, for the remainder, the remaining few weeks of this course, is it going to be that we're mainly talking about culturing meats, or will it be like, is there going to be a time where we talk more about the precision fermentation, or is it just the fact that like it's such a relatively underdeveloped field that there's not a lot to talk about yet?
Yeah, I would say that since it's an underdeveloped field, it is going to be a bit more difficult for us to have discussion about it. And we're going to be talking about sort of both as they're relevant. Yeah, but we're going to try to make it about 50-50 as much as we can for cultured meat and precision fermentation. Yeah. So I did just share these slides so that you can take a look at the content for them, but also because I have a couple of things for you all to do now. So for your homework for today, I would like you to read The Scientific Sustainability and Regulatory Challenges of Cultured Meat, which was published earlier this year by some of the biggest names in the field. And this should give you a really good overview, help cement some of the things that we talked about in the crash course, and make you think about the final project's ideas. But for right now, I'd like you to all pull up this class survey that we have. It's really short, and it'll just give us a sense of who's here, what are you interested in. Can everyone find the intro survey? Can you give me a thumbs up reaction from the participants? Awesome. Looks like this. Let me know when you're all done. I will say 5 minutes.
Sorry, where is, oh, okay. I didn't see a link. Thank you.
Oh, I think you can also put it in the chat.
I'm sorry, I have a question about the how to translate my Italian degree to the the American format.
Yeah.
I have no idea. I'm always so confused when I try to look it up on the internet. So I did 3 years of university. So is that considered a bachelor for you or no? Because in Italy it's very linear. You have 3 years and then you can do other 2 years, and that's a master, kind of.
I mean, for us in the US, our bachelor's is typically 4 years, but
Okay.
that was one of the reasons why I wrote that question not as multiple choice, but just write in
Okay.
what makes most sense. I don't want to confuse you. Let's see. See about 10 responses so far, so half and half. Give you 2 more minutes. Is there any question tripping people up? I hope the survey was understandable. All right. Thank you, John, for coming.
Yeah.
Okay, waiting for response for I think one last person. Oh, okay, no, we're good. Yeah, so I think we can get started again. So now I want to talk about our final project. As I said before, there are 3 options. You can do a homemade fermentation recipe, a variation on an existing paper, or a project proposal for a UROP. And if you go to this link, you can find the resources that we've accumulated to help you all with your final project, whatever you choose. Awesome. Yeah, so here we have some papers that we think that you all could actually just do in your homes. So the decellularized leaf scaffold is actually pretty home friendly. There's some other scaffolding that you can make. There's also Shojin Meat, which is Shojin Meat's manual. So Shojin Meat is actually communal biomaker space. And they sort of specialize in, they've developed a lot of protocols for how to make your own little bioreactor or grow your own cells at home. I wouldn't suggest eating these though. You know, just there's not any food safety checking that you can really do. But it would still be super cool if you did it. Yeah. And then we've also come up with a list of suppliers for ingredients. So Cultures for Health would be useful for any sort of fermentation that you have. You can basically buy cultures there for sourdough, kombucha, yogurt, anything you want. Modernist Pantry also has things like calcium hydroxide that could be useful for the decellularization scaffold. Can you share the Google Drive link? Yes. It should be, you should be able to find it in the slides. I hope that you can find the slides, but I can share the Google Drive link. Okay, so that's suppliers. Then we have linked resources. So these are books and collections of papers that you could find useful. So I can probably find some way to get the ebooks to non-MIT students if you would like that. So we have ordered Sandra Katz's The Art of Fermentation and Renee Redzepi and David Zilber's The NOMA Guide to Fermentation from the MIT library. So you should be able to find those for free if you are an MIT student. But perhaps email me if you are a non-MIT student and you would like access to these resources and you can't get them at your school. Then there's this paper pile of cultured meat papers developed by Elliot Schwartz. Unfortunately, he hasn't done one for precision fermentation, but this is a super useful resource separated by subfolders on anything that you could want, religious considerations to scaffolds. I've noticed that he doesn't have all of the papers relevant for consumer acceptance, but you could look up Cor Vanderweel for lots of good work on consumer acceptance and more social science side of culture and meat. Yeah, unfortunately, I don't have much to share for precision fermentation papers. There's just not a lot available, as I found.
Yeah.
Yeah.
I would just say that for precision fermentation, as I mentioned in the crash course, probably one of the best guides out there is the Good Food Institute report, which is linked in the crash course slides.
Yeah. It's the older field, but in terms of like there's been the food enzymes and insulin that and Renee that have been around for a long time, but there's not a lot of papers on the specific area of having these ingredients be for food. Yeah. So then I want to talk about our final project ideas. So we really hope that as many of you as possible, if you're MIT undergrads, can think about making a project proposal. Also, if you're not an MIT undergrad, think about a proposal for a grad student project or a master's degree. And I used to work at New Harvest, so I can actually connect you to a potential funding source. If you are looking for graduate funding in cellular agriculture, it would be super exciting to have you do that. So for the final project proposal for a UROP or a different research project outside of MIT, we ask that you suggest 3 to 5 PIs at MIT who would be good supervisors to this project, and we'll help you email the professors and find funding for your UROP as necessary, or your research program, as I've worked for New Harvest before. And then we want to give one example of what a project proposal could look like. So we want you to have a title, a list of potential investigators, and why you think that their work could be relevant to this topic. So this can be, if this is an MIT Europe, you'd probably want MIT professors, but if you're in a different university, then I would suggest finding professors that are in your university and that you can go talk to. Then we want you to give a bit of background on the area. We want you to give a little bit of background. Sorry, I'm getting Slack messages on the topic as sort of like a little mini review of what's been done. Talk about your objectives, the variables that you'd like to cover in your experiments, and your materials, methods, and relevant research papers. Okay. So does anyone have any questions on the final project? Or we can help you get started trying to find these resources. It's going to be like more you time for the next few minutes.
So if I want to make a homemade recipe, I mean, I just do it, and then on the last day we show the yogurt or whatever it is that I've created to the class.
Yeah,
okay.
basically that. But I think it would still be cool if you could present a little bit on the science of that particular.
Okay, sure.
Yeah. So how does what is the bacteria involved? What do they do to the yogurt to change it from milk to this delicious creamy substance? Yes, let us know a little bit about the science.
Okay, so also a kind of a presentation. Okay.
Yeah. Does anyone have ideas for a project proposal or areas that they think are interesting that I could point them in the direction of? Yeah, all right, so I'll be here for the next 20 minutes in case you have any questions or if I can help you with your proposals. But this can be more you time if needed. And I will be showing you how to use the MIT libraries here to find the NOMA guide to fermentation. It's just an example.
Also, I had a quick question. I think that would people be interested in having like an office hours to potentially like do maybe a one-on-one or something, talk about project ideas or area they're interested in? You just like say in the chat if that's something you like or say.
Yeah, we could definitely do that.